Hi everyone. Hello, how are you doing? I hope you're doing good. So welcome to another chapter, which is new chapter, chapter 12, biotechnology. Now you are already aware of what biotechnology is because you have done this in lower standard also. Basically, when we talk about biotechnology, it is nothing but interaction between biological science and technology, right? Basically, it's an applied branch of biology. You can say that biotechnology basically was coined by this term was coined by scientist Carl Ehrlich. Okay. So in this chapter, we are going to deal about what is biotechnology, what are the different processes, applications, and principles of biotechnology. Okay. Now, in this video, we will be mainly talking about the principles of biotechnology. Now, there is two, when you talk about biotechnology, so one is the age-old traditional means of biotechnology that we have covered in the previous chapter also, and the another branch is the modern biotechnology. Okay, wherein you have new techniques, for example, fingerprinting, then for example genetically modified crops or genetically modified organisms, and traditional ones which we covered in the previous chapter, wherein there is use of microorganism in the preparation of curd, ghee, vinegar, yogurt, and all those stuff. Okay, which has been in tradition, in practice till age old time. Okay, at that time, people did not know as to what exactly is the process occurs and the organisms causing this process. But over a period of time, with the contribution made by several chemists, biochemists, and microbiology, they could explain the mechanism and the process of all the nature of the microorganism which were causing these biological processes. Okay. And then lately, in 1970, a new technique known as, recombinant DNA technology was developed by scientists, which, changed the overall outlook of what biotechnology is. Okay. So basically, combination of biology and production technology, okay, based on mostly genetic engineering. So together, they form the modern branch of biotechnology. Okay. Yes, so now we begin with the main principles of what is biotechnology. Now, modern biotechnology, when we talk about it, is having two techniques mainly: genetic engineering and chemical engineering. Okay, one is genetic engineering and one is chemical engineering. So obviously, we will be focusing more on the genetic one because we are focusing on the biology. We are dealing with the biology subject. So we will be discussing about the genetic engineering of different, with respect to biotechnology. So when we talk about genetic engineering, what are we talking about? We basically, genetic engineering deals with you are going at the gene level, you are altering the genetic material, either the DNA or the RNA, okay, of any organisms. You are altering the genome or the genetic material of that particular organism, whereas chemical is basically dealing with maintaining the sterile environment for manufacturing various useful products like vaccines, like antibiotics, enzymes, organic acids, vitamins, etc. Okay, so genetic engineering is manipulating, is causing alteration in the DNA or the RNA, okay, towards, why are you doing this? So that something you are desiring for, you get. Okay, you are expecting an outcome which you are desiring. Yes, so that is what genetic engineering is. You are making an alteration in the gene level so that whatever outcome, whatever result you are expecting, that is according to your desire. Fine. Okay, so moving forward, so let's define technically what is genetic engineering. Uh, so generally, when we talk about biotechnology, it is dealing with techniques using live organisms or enzymes from microorganisms to produce and process useful things for the humans. Okay, and the main aspect of it is genetic engineering. One of the major example you have studied in schools also, that is the IVF process. Okay, the in vitro fertilization of the egg and the sperm natural setup. Okay, sorry, in artificial setup, in a lab setup, which is also known as very commonly test tube baby. wherein so this is one of the major example of biotechnology. Then either you are doing synthesizing a gene and using it, or you are developing a vaccine, or you are correcting a defective gene if any person is suffering from a genetic disease. So you go at the gene level and you correct the gene, and then chances of the person, getting treated with that particular disease is more. So all this is part of biotechnology. Okay. So moving to the principles of biotechnology, as we have just mentioned, the major is the genetic engineering, also chemical engineering, but chemical engineering is mostly when we talk about the processes and the steps involved in, manufacturing of useful products like vaccines and vitamins and minerals, etc. But whereas genetic engineering is you are applying techniques, you are applying processes to alter the genetic material, to alter the chemistry of the genetic material, to introduce these into new host and organism, and depending upon the altered DNA and RNA or genetic material, now you will get the phenotype as per your desired outcome. Okay, so that is what genetic engineering is all about, manipulating on the basis of what you desire. Okay. Also, then the chemical engineering is basically maintenance of the sterile ambience in chemical processes to enable growth of only desired microorganism, or if you are processing a cell, eukaryotic cell, so in large quantities in big fermenters for manufacturing of biotechnological products like antibiotics, vaccines, enzymes, etc., etc. Okay, so these are the two main branches of modern biotechnology. Now we begin with the first principle, that is the tools that you require for the basic for biotechnology, which involves three basic important steps in genetically modifying any living organisms. Okay. So the first step is, identification of the DNA with desirable genes. First, you identify which gene are you interested in. Yes, so you identify that, as you see in this first figure. Okay. So let's say out of the whole genome of any organism, you are interested in this gene. Then you isolate that gene, you extract the DNA. Okay. Now you will require any host organism or a living organism in which you will insert this desired gene, so that when this host can be a eukaryotic cell or a prokaryotic cell. Okay. Eukaryotic cell or a prokaryotic cell, like any bacteria. So this desired gene, you will have to fix it somewhere, so that when the living organism is multiplying, so accordingly it will make copies of this gene also, and this gene will produce some product, some outcome, so that also will get multiplied. So that this is talking about the second step, that is introduction of the identified DNA into the host, and then inserting this DNA into the host. Okay, and maintaining its progeny, maintenance of the introduced DNA in the host, and transfer of this DNA to its progeny, and then you scale up to a bigger at a bigger level. So that is how you get genetically modified crops. So this is at at the lab level, and then you take these cells and then you grow it on a larger level. Okay. So this is in general we can call it as gene cloning. You take this single gene and then you multiply it with the help of the host organism, a living organism, and you get multiple copies of the desired gene. So a gene of the known function is transferred from the normal location into the cell. Okay, we can call this as the vector, the carrier cell or the vehicle. And then the transferred gene is replicated normally and is handed over to the next progeny. And then now this is embedded in the next progeny. So if the plants multiply, so automatically the desired gene that you are looking for will be multiplied naturally. Okay. So this is what a general understanding of gene cloning, or you can say, recombinant DNA technology is all about. Okay. You can call gene cloning or you can call recombinant DNA technology. They are interchangeably, alternatively, they are used. Yes. Moving forward. what are the different tools and techniques? Okay, what are the different raw materials, tools that you require for this gene cloning process or for this recombined DNA technology? Yes, the first is, obviously you will require the enzymes. Now, what is enzyme? In the previous figure, we saw this is the cell, this is the nucleus, you have the genetic material inside, and the genetic material is very, very big, very large. So you require only, let's say, a short segment of the DNA which you are interested in for producing your desired protein, let's say, for example. So out of this whole length of DNA, you require only the small part of the DNA. So for this small part of the DNA, you require scissors to cut this small part of the DNA and take it out of the entire genome. So this scissors is known as restriction enzyme. This is the very, very important tool in gene cloning process. The restriction enzyme, or you call them as scissors. Now the name itself tells you restriction. They will cut the DNA at specific points. Specific means restricted site. So the DNA that you want to cut, they have a particular sequence, right? And the enzyme will also have a complementary sequence so that it goes and bind to that target site only. It will not go and bind to any other place in the DNA. It will go and bind to the target site only. So it will restrict itself to this specific binding, and it will bring about a cut in the DNA. Okay. Yes, so cuts at specific point. Then it also cuts vector and foreign DNA. So it will cut this restriction enzyme has a property of cutting any type of DNA. Either the DNA is of your foreign origin, or sorry, human origin, or any plasmid origin. Plasmid mean those extra chromosomal DNA which are generally found in bacteria. Okay, so they have this property of cutting this diverse amount of DNA. Cleaving DNA will make what? DNA fragments. So it will cut the DNA into smaller fragments, which will result into this single-stranded segments. As you see in the figure, for example, restriction of a restriction enzyme, the enzyme cuts, okay, this is the foreign DNA and this is the vector DNA. Let's say the enzyme binds over here, the arrow that can be shown. So, in the vector DNA, it cuts at this site like this. A staggered cut is made, and in the foreign DNA, it is cut like this. Foreign DNA means this part of the gene you want in your vector DNA. This part of the gene you are interested in combining with the vehicle, that is the host DNA, the plasmid DNA. And then it will bind, it is cut in such a sense that it creates this hanging nucleotides, so that after the cut, the DNA fragments will automatically join one another due to complementary base pairing. And then this is how you get a recombined DNA. Your desired gene and the remaining genes are of the carrier DNA, that is the DNA of the host, either the plasmid or the genome of any bacteria. Okay, so the sticky strands, this setup is known as sticky end. So the sticky end will allow insertion of the foreign DNA into the vector DNA. Okay, and double-stranded current will produce blunt end. Okay, so sometimes this type of cut is created, sometimes direct cut is created, like this is the blunt end, or sometimes this type of cut is created, creating sticky ends. Okay, so very, very important tool are the restriction enzymes. Yes, they are nuclease in nature. They can be either exonucleases or they can be either endonucleases, right? So exonucleases will cut at the end of any DNA. Exo, the name itself is telling you, exo will cut at the ends of the DNA molecule, whereas endo will go and bind in between any place on the DNA, and it will cut in between. Okay. That is in the middle of the DNA molecule. So basically the phosphodiester backbone, they are cut by these restriction enzymes, and depending upon, they are giving, depending upon which bacteria they are coming from which bacteria they were isolated, they have different names. Okay, they have different names to it. Yes, because they are used by the bacteria to destroy various viral DNA, and that might enter the cell, thereby, restricting the potential growth of the virus. So, these restriction enzymes basically serve as a defense mechanism in the original bacteria in which they were found, but they are then used for this cutting purposes and different bacteria. Okay. So this restriction enzymes are basically molecular scissors. They will do what? They will go and recognize the DNA at specific places, at specific sequences. These sequences are known as recognition sequence. What are they known as? They are known as recognition sequence. So different enzyme found in different organisms will recognize this different nucleotide sequences, and therefore they will cut the DNA at different site. Okay, so these are the examples of some of the enzymes, isolated from some of the microorganisms which can recognize these sequences. So this sequence found in any organism can be cut by this enzyme. Again, the sequence found by in any organism, plant genome, animal genome, any other bacteria genome, can be cut by this enzyme EcoR1, which was originally found, which is taken from this bacteria Escherichia coli. Okay, they can actually, these sequences recognized by restriction enzymes are usually 4 to 8 nucleotides, as you see in the table also, 4 to 8 approximately. Yeah, so a sequence is there. Sometimes, sometimes the sequence will be palindromic in nature. Palindromic means, as you can see over here also, yes, for example, it is like, A, A, G, C, T, T, and on the complementary side, it will be exactly opposite of this. So T, T, C, G, A, A. As you see, mirror images of each other. So this is also known as a palindrome. This type of symmetry is known as a palindromic symmetry. Okay, so the sequences which are running in this direction, the same sequences are running in the opposite direction. Yeah, so they will cut the DNA and produce either sticky ends or the blunt ends depending upon the cut. Fine. So this is one of the important and major tools in the recombinant or DNA technology or the gene cloning. The next very important tool is the cloning vectors. Okay, very, very important. We were talking about the desired gene taken, and then they are inserted into what? They are inserted into the host genome. Okay, so host genome, so that they can, embed into the host genome any bacterial genome. So as and when the bacteria is also multiplying, the desired gene that you inserted into the genome of the bacteria, that will also multiply. Okay, so they are known as cloning vectors. So vectors are again DNA molecules that will carry a foreign DNA segment, and they will replicate inside the host cell. It can be plasmid, which are circular extra chromosomal DNA of the bacteria. This we have discussed in Levin standard and your lower standard. Also, viruses which infect bacteria, bacteriophages. Okay, many examples are there of chloric vectors. Yeah. So two examples you can see PBR322 and TI plasmid. You have these sites, different sequences. Okay, this is the origin of replication, and these sequences are named according to the origin and which sequences do they recognize. Yes. So these are the example. For example, a plasmid can have a carrying capacity of 0.1 to 10 kilobase pairs. Okay, it has a marker gene of ampicillin and tetracycline resistant gene. Basically, this plasmid is used for the bacteria which it resides in. It is, capable of protecting that bacteria from this antibiotic ampicillin and tetracycline, hence resistant gene. Then you have bacterial artificial chromosome. These are some of the examples of the cloning vectors. Okay. it can carry more amount of your desired DNA. It is here it can carry 35 to 300 kilo base pairs, and it has marker for this antibiotic, chloramphenicol and lactose metabolizing gene. Okay. Modification of the F plasmid, which is required in copulation process of a female and a male bacteria, and it is artificially synthesized. Example is over here. So you need to know the examples basically, nothing much. Okay. Another is yeast artificial chromosome. It has the carrying capacity of 11400. Okay, they are used to they are used to clone DNA fragments of more than 1 million base pairs. Okay. So basically, as you see, as we are going down, the carrying capacity increases. So it's like this is a bicycle, this is an auto rickshaw, this is a car. So depending upon the size of the vehicle, those many passengers can be boarded onto that vehicle. Okay. And finally, you have the cosmid, which can carry 30 to 50 kilo base pairs of your desired gene. So plasmid is having similar properties to a phage, that is a virus that infects a bacteria, and a plasmid, hence the name cosmid. An example is COS1. You just need to know the examples and the name of it. Okay. And also you have plasmid vectors. Compound is plasmid and a phage. So plasmid and together a phage together will give you a plasmid, which is a structure made of bacteriophage and a plasmid. They are used for carrying larger DNA sequences. So depending upon what your desired gene is, how much long it is, you can choose which vehicle you want to consider. Also, very important properties that a vector should have is it should be small. The vehicle should be short and small. It should be compatible with the host cell. That is the vehicle, the host that you will be, the vector you will be inserting into the machine, living machine, that is the bacterial cell. So that bacterial cell should accept that vector. Otherwise, if it recognizes foreign, then your desired gene contained in the vector will not be able to multiply. Okay. It should not be incompatible with other vectors. It should be incompatible with other vectors. So it should be very specific. Should be high copy in number. Means what? Your vector should be able to multiply and give you more number of copies. Okay. It should be able to express itself in utilizing the host machinery. So that is what compatibility is all about. The host cell, should allow this inserted foreign DNA along with the vector to express itself, means to carry out transcription and translation. So the host cell should provide all the machinery, all the enzymes, all the raw materials, all the energy for this replication process. Okay. And it should be able to move under two systems. So it should be it can move from it should be compatible with the prokaryotic as well as eukaryotic system also. Okay. Two examples that we can see in the coming picture is common that is used in plant and animals. So Agrobacterium tumefaciens, this is very, very commonly and very important host cell. Okay. This bacteria is capable of carrying. So this Agrobacterium tumefaciens, which is basically a soil bacteria, it is capable and it is very good in carrying this tDNA, transfer DNA, to transform normal plant cells into the tumor and direct these tumor cells to produce chemicals required by pathogen. Meaning what? This Agrobacterium tumefaciens, it is having this plasmid. So this bacteria is capable of carrying this plasmid, and this plasmid causes tumors in the plant cell, in plant. Okay. So if you see, this is a normal plant, and if this bacteria infects another healthy plant, it can cause tumors, crown gall. Okay. Now genetic engineering has manipulated this Agrobacterium tumefaciens. Okay. It has manipulated Agrobacterium tumefaciens. It has, taken this tDNA, Ti plasmid. This plasmid is responsible for causing these tumors in normal healthy plants if the bacteria infects this healthy plant. So what bacteria biotechnology has does is the TI plasmid is modified genetically, means the disease causing segment, the tumor causing segment is removed. This is the TI plasmid, and this segment is causing disease. So this is removed, and in this place, what is inserted? Your desired gene can be inserted. Anything. Let's say you want a good quality crops, or you want disease resistant crop to a particular specific disease, or you want good yield crop. So your desired gene can be inserted in this place, wherein the pathogenic gene is removed, and then this can be inserted into the bacteria, and then this bacteria is naturally capable of infecting, or moving to plants. So this proves a good vehicle, a good vector. Yes, so TI plasmid is modified into a cloning vector which is no more pathogenic to the plants. So this is one of the very most commonly and easiest vector used. Another is, retroviruses in animals that can be used. They have the ability to transform normal cells into the cancerous cells. We are very much aware the retroviruses, the ones which carry the RNA as a genetic material. So what can be done? The RNA, the genetic material can be cloned, the pathogenicity can be removed, and the desired gene can be inserted, and natural tendency of the virus is to infect the host cell. So if it is infecting, no problem, because we have removed the pathogenicity part and we have inserted our desired gene part to it. Okay. So now they are manipulated, they are engineered, disarmed, and now they are used to deliver desirable genes into animal cell. Understanding? So some of the nicknames of biotechnology that you should be, knowing just for remembrance. For example, plasmid, as we are calling it as vehicle. So it is basically the gene taxi. As a taxi, you take a taxi, you have to go from one place to the another place. So like a vehicle, like a travel vehicle. Then the actual thing, that is the workhouse of the gene cloning, that is the thing which will produce multiple copies of a desired gene, is your plasmid. Okay, again, artificial plasmid. Molecular scissors are what? Restriction endonuclease. Then again, cutting desired sites of the DNA is your restriction endonuclease. The ones which will join the remaining gaps will be the enzyme ligase. Okay, chemical knives, restriction endonuclease. Natural genetic engineer, which can naturally, infect plants and can be used for genetic purposes, agrobacterium tumefaciens. Okay, mobile genetic elements. These are certain sequences of the gene which can, move from one place to the another, jump from one place to the another place. Place means what? From one place in the DNA to another place in the DNA. So they are known as transposition, transposons. Okay, passenger DNA, that is, your desired gene that you want to be carried to a specific place or specific plant. So that will be your foreign DNA. Okay. So that's all with regards to this video. We have discussed all the principles of biotechnology. Now in the next chapter, we will be talking about the how this process is done in detail. Okay. Right now we have understood what are the tools that are required. So you need a scissor, you need a cloning a host cell, you need a cloning vector. Okay, and what are the qualities of the vector that should be kept in mind? So that all we have discussed. Now in the next video, we will be talking about the steps which are involved in recombinant DNA technology. So thank you, take care, see you, bye.